Hi everyone, I'm Anid.
And I'm Chen Qian, and we are here to present our final project for EC523. We attempt to write a deep learning algorithm that will select the correct location of neuron for calcium image analysis. So calcium imaging is a common tool that neuroscience use to monitor the activity of hundreds of neurons. So basically, we already know that when neuron became active, it have the members potential increase, and there's a great influx of calcium ions. So we just transfect a genetically encoded calcium sensor GCAM into the neuron. Then we could just observe the fluorescence of that GCAM to show that whether a neuron is active or not. So you could see that they are blinking of punctures, and in the background, this is because that neuron is like a fire. It fires during when the mouse is running. So this is actually a in vivo recording. So our job is to basically select the location of all those punctures because we want to extract the trace, the average calcium fluorescence throughout the recording section. So basically, this means that we need to draw hundred of circle around this, like a bright dot, which is really time consuming. That's why we want to write this code that do that automatically. Okay, so here's a more close-up look. So we basically before we draw this circle by hand and we run it, we run it through just a simple MATLAB for loop that extract all the trace information here and do some background subtraction, and we got this heat map, this heat map which will present all the neuron, how all the neuron basically their activity within the recording section. This is how we do calcium image analysis. Next, for this project we use the data collected by Sudicus in Hans lab. She did a 26 set of data this 26 set of imaging, and we will each imaging there comes a field of view and a label, which are all handpicked. Then the big imaging is 1,000 by 1,000 pixel. So we will use 23 for the top 23 set for training and the bottom 3 set for testing. So, well, 1,000 by 1,000 is too large for our network, so we will cut it down to 256 by 256. And we will cut 1,000 small image from a single large image because 26 is not enough for deep learning training, and we will make it a total of, for the training it will be 23,000 set of image and label, and we will just cut 10 out of the last 3 big image, 100 out of the last 3 images, so we will get 300 testing data. And yeah, this is how we train our network. So we choose to use the UNet architecture because it is the most common architecture for a biomedical image segmentation. So it's basically the downsampling part is really similar to just the CNN, and because you have convolution network like a max pooling, and but the up one is just interpolation, which doesn't require any more parameter, and we use the concatenation between the down part and the end and the up sampling part. So basically, this information will communicate throughout here. So basically, we will get our input image and scale it down in dimension and rise it up. So at the end, we will have the same dimension as our input, and we hope that we will implement a softmax function at the end too. So basically, we will do a binary classification for every pixel, that whether it belongs to the background or it belongs to the ROI within the circle. And we will also implement a UNext++, which is just a fancy version of the UNext, but we have convolution, like a bridge of convolution within the down sampling and up sampling part, to allow further communication between these two. And it contains more parameter and it's slower to train, but it may produce a better result.
Okay, so before we can train these networks, we have to do some pre-processing on the data, mainly because there's some irregular noises that we've seen. So we did this with some basic image processing techniques such as normalization, mapping to grayscale, and histogram equalization. And then these images are just showing how before the histogram equalization, the images are dull, low contrast, and it's difficult to see the entirety of the neurons. And then after applying the histogram equalization, the images are less distorted, and we also saw that we get better accuracy in our model. So a little bit about the network settings and metrics. So, so the training data size again is 23,000 images. The testing data size is 300 images. We trained for a total of 3 hours, which is about 20 epochs, with a batch size of 16 and stochastic gradient descent using Atom Optimizer with this learning rate. For metrics, we use 3 different metrics. The first one was pixel accuracy, which is just the percentage of pixels that are correctly classified as either background or ROI. And this is really good for binary labels that we have. And then the second metric is the intersection over union, or IOU. And this is simply just the area of overlap between 2 objects divided by the area of union. The third and last metric we use is the dice coefficient. So this is twice the area of overlap divided by the total number of pixels being occupied by these 2 objects. So one issue that we noticed in our model is that there's a lot of overlap between regions of interest, and then we think that that's causing some inaccuracies for our model. So what I'm showing here is just a representation of what we see. So because we have manually selected labels, a human selected these labels, we have a lot of ROI overlap. And then that looks something like this. So what we did to segment these is use what's called the watershed segmentation algorithm. This is just a very traditional segmentation algorithm that, given 2 objects, it does some sort of topological mapping, and then it's able to segment these 2 objects like shown here on the right. So we can apply this watershed algorithm to the predicted labels from the network. So you can see here that there's a lot of overlapping labels. And then if you apply this algorithm, we can see that these labels that were once overlapping are now segmented. So getting into our results now, here on the left are the results for UNet, and on the right are for UNet++. So you can see that after the 20 epochs, our loss generally seems like it's going to converge, and the 3 metrics all seem to be pretty high and also converging. Same goes for UNet++. The only difference with UNet++ is the loss was slightly lower at the end of the training.
Also, this loss is like cross entropy dice law. It's the one we define based on the dice coefficient. Yeah.
So here is just one sample image and label. So here are the original labels, and then here are the predicted labels. So what you can see here is that the predicted labels capture pretty much all the neurons. And then the ones that are overlapping originally, those can be segmented using the watershed algorithm. And then here on the right, we're just showing the multi-class labels, which just qualitatively gives us an idea of all the neurons that were predicted. And we can also look at the centroids more nicely. Okay, so lastly, we can also look at the overlap of the centroids between the original and predicted labels. Here's just one example from one sample. Here in this example, the mean distance between the matching centroids is 0.65 pixels. And then across all testing data, it was about 0.85. And then also importantly, we're not able to predict all of the original labels. There's some that we failed to capture, some that you can see right here.
Yeah, so here the plot twist. So the previous data looked really nice, and actually, like, it is the result like after trained of like 26,000 data and use the validation set, which is like part of the training set, which means that all the previous results is actually coming from the training data. And when we realized this mistake, it's like just the day before yesterday, and we redo the training and realized that there's a serious severe overfitting problem. So here, as you could see, like the blue curve all converge, but the orange testing loss and all the things it just stuck, especially the dice coefficient just stuck just between 0.5 and 0.6. And as you could also see in those graphs, like the performance on training set is much better than testing set. And the UNet++ doesn't really solve this problem. It's almost the same result. And so this is really unfortunate. So we are blinded by our like false goods result, and then we failed to improve it or find the true problem. Like so for the past few weeks. Yeah, I think like there are some problem with this. The main cause of overfitting is like during our data pre-processing, like there's a normalization occur, and this normalization, like when we select a box, like on the dark region of the raw image, this over normalization will be seen like distort the original content. Like there are some neurons that are like really dark, it's almost blending into the background now. So when you scale it up from 0 to 255, it became indistinguishable, but the network will treat it as the same as other box around the bright neuron. So this will cause the network to learn the feature that does not exist in the first case. Also, like we cut 1,000 small image from a single large one, so there are some features that will be repeating. So the network is actually learning repeating feature over and over again. Also, that we just don't have enough data because we only got 26 large image and we only train on 23 large image. Yeah.
Okay, so in conclusion, so we found that the simplified UNet we implemented suffers from overfitting, but it's likely that this problem can be solved by getting more data or better preprocessing, such as just better data augmentation that goes further than just cutting up the images. More data can be available from Dr. Han's lab, which Cheng works in, so we could always further improve this model by getting more data. And then lastly, something like this could definitely be used by researchers for neuronal segmentation of calcium imaging data, given that we're able to further train it. Thank you.